everyone. My name is Meena Ramchandani. I'm an infectious disease physician at the University of Washington in Seattle. This podcast is dedicated to an STI review for healthcare professionals who are interested in remaining up to date on the diagnosis, management, and prevention of STIs. In this episode, we welcome back Dr. Tara Reid from the University of Washington. Dr. Reid's research focuses on bacterial pathogenesis and T-cell immunology, specifically looking at the immune responses to syphilis. Welcome, Tara. It's great to have you on this episode again.
Thank you.
Let's talk about an article that you published with colleagues in Journal of Infectious Diseases in August of 2024. For our audience, it's titled Treponema pallidum, Periplasmic and Membrane Proteins are Recognized by Circulating and Skin CD4 T cells. Tell us a little bit about why you did this study and what was your hypothesis?
So there's a lot of data, a lot of immunohistochemistry data and microscopy that shows that there's a CD4 infiltrate in early syphilis lesions. So we know this. We also know that people who are infected with HIV who have really low CD4 counts can have really severe presentations of syphilis infection. And so we knew that CD4 T cells were important. We also know from the rabbits that splenocyte responses, including CD4 T cells, are important during infection. But what we didn't know was what antigens the CD4 T cells are recognizing, what bacterial antigens they're recognizing to stimulate their effector functions. So we sought to identify those antigens.
And you enrolled 4 cohorts in this study. Tell us a little bit about the different cohorts and why did you choose these different participants?
So this is really important. We initially started out by enrolling people with secondary syphilis. So these are people that have symptomatic syphilis. We can see their syphilis. We also looked at people with latent syphilis, including early latent and late latent. So these are people that clinically we think are infected with Treponema pallidum, but for whatever reason, they don't have outward signs of the infection. They don't have lesions. Thirdly, we enrolled people that don't have syphilis at all right now, but they've had syphilis in the past. So they've been treated for syphilis in the past. This is a nice group to study because whatever immune responses we detect in them against Treponema pallidum, those are more durable responses because they don't actually have an infection right now. Maybe they were treated 5-10 years ago. And then lastly, as with most of these kind of studies, we like to enroll people and study folks that have never been exposed to syphilis and they have negative RPRs, negative treponemal tests as well. I'll add, though, that we did not include people with primary syphilis initially because we wanted to test for adaptive immune responses, and typically those take maybe a few weeks to develop. I think that we are currently of the mindset that we might go back and do these studies also in people with primary syphilis to see if we're able to identify CD4 T cell responses in that group as well.
What was the average RPR in the cohorts that you studied?
So amongst the cohort with secondary syphilis, we measured RPRs ranging from 1 to 16 up to 1 to 512. So the average was maybe 1 to 128. In our group of participants with latent syphilis, again, that includes early latent and late latent, we had RPRs range from non-reactive up to 1 to 256. So we measured or kind of guesstimated the average there to be 1 to 16. And I'll note that the participants with a non-reactive RPR actually had a positive treponemal test.
Did you study persons with HIV and what was their average CD4 count?
We did include people living with HIV. I think there were 8 in total in this study, and their average CD4 count across all groups was about 686. I'll note, though, that the minimum CD4 count that we measured in our participants living with HIV was, I think, 312, but we've had people that had a CD4 count greater than 1,000. So in general, I would say that in this cohort of people, they weren't terribly immunosuppressed at all.
But there was quite a range, and it sounds like everybody was on antiretroviral therapy, right?
Everybody was on ARVs, correct.
Tell us a little bit about how you identified T. pallidum-specific T cells.
So I was lucky in that I had a great example of tools to use to find pathogen-specific T cells. So David Cole has used these pathways to study and identify T cell responses to different viral proteins during herpes infections. We used those same experimental protocols and applied them to syphilis. In summary, I'll say that from PBMCs, which is the first tissue type that we studied, we take the entirety of the PBMCs from a blood sample, stimulate them with whole Treponema pallidum antigen. So we take the bacteria and smash it up and use that to stimulate these cells in a petri dish outside of the body, and then measure the activated CD4 T cells. So these are the T cells that get stimulated by the Treponema proteins. We can then sort those activated CD4 T cells. So you're separating those from the ones that aren't activated. So these T. pallidum specific CD4 T cells, we can sort and then grow them in culture. So now we have millions and millions and millions of these cells that we can use for downstream studies.
And so these are finding these CD4 T cells from the blood, correct?
Correct.
And for our audience, what does PBMC stand for?
PBMCs are the peripheral blood mononuclear cells. So these are your immune cells that are circulating in the blood. We can take the blood and separate it into different fractions. So you have your red blood cells, you have your plasma, and then there's a small fraction that are these white blood cells that we can separate from the rest of the blood.
So tell us a little bit about the interferon gamma response and why you chose this measure.
The T cell response that we see in tissues is Th1 polarized. So what that means is there's a CD4 T cell subtype that produces interferon gamma in response to stimulation through its antigen receptor. So we used interferon gamma as a measure of activation for these CD4 T cells. We also use IL-2 or interleukin 2. This is another effector molecule produced by these Th1 CD4 T cells. In addition to those two cytokines, in vitro in these assays, we measured proliferation as a measurement of how activated these cells were, or if they were proliferating in response to different Treponema pallidum antigens.
That's helpful. So you would have the CD4 T cells that you sorted out and then enriched, and then you would put them together with the Treponema pallidum specific proteins, the different proteins, let them cuddle together, and then measure the interferon gamma response and the IL-2 response and proliferation.
Exactly. I like cuddling.
And tell us a little bit more about enriching and expanding the T cells. Why was this done?
Yeah, that's a great question. So in your peripheral blood mononuclear cells, there's T cells and other immune cells that are responsive to so many different antigens that we encounter day to day, even antigens and pathogens that we've encountered throughout our lifetime. So in this complex milieu of different cellular T cells and B cells, we wanted to find just the ones that were responsive to Treponema pallidum. When we measure those, using the assays that I mentioned before, when we measure those cells can be quite rare. So amongst all of the PBMCs, all the CD4 T cells, they could be as low as less than 0.01%. That's incredibly rare. But we start off with millions and millions of cells. Even if we sort and find 100 or 1,000 cells, we can then use those cells to answer these questions. But it's such a small number, right? Only 100 cells. How do you do experiments with such a few number? So that's why we expand them. So we just grow them out in culture. So then we have millions to play with. and ask these questions.
Did you find that people who had, let's say, secondary syphilis had more T cells than those who had, let's say, latent syphilis? Or was there not a difference?
Not necessarily. And it's hard to make that direct comparison based on how much blood volume you take from a person, how efficient your isolating of PBMCs was, and then additionally, how many cells you start with as you're stimulating with T pallidum and then getting the treponema pallidum specific CD4 T cells out. So we didn't measure that specifically. We were more interested in how many we could get expanded. Mm-hmm. I'll also add that when we take these treponema pallidum specific T cells out of the PBMCs or out of the blood and then expand them, it's a polyclonal expansion, right? So there's a lot of different types of cells there that could be responding to different treponema pallidum antigens. But it's important to think about because it's not just one CD4 T cell that's responding to one antigen or maybe even a few. It could be hundreds of different T cells that are responsive to 100 antigens. It could also be 100 different CD4 T cells that are very closely related and they're sisters and they're all clones of each other or near clones and they're all responding to the same antigen. That's another scenario. So by measuring that absolute number, it doesn't necessarily give us any more detail about the breadth of the CD4 immune response for an individual.
There's a lot of variables.
Mm-hmm.
And were you looking at also tissue-resident memory cells? And what were your findings? So you got some from the PBMCs in the blood, but what about the tissue?
We were excited to investigate what was going on, again, at that interface between the bacteria and the immune system, right? So by looking in tissues, we're able to do that. We use a different protocol to answer these questions. So I mentioned that these treponema pallidum specific CD4 T cells can be quite rare in the entire blood volume, right? It could be very, very rare. So we have to expand them. We have to separate them and expand them. It's a different story in tissue, though. So within skin biopsies, there's bacteria present. There's treponemes present. We can stain them. We can see them. So there's antigen there. Because there's antigen there, we think that there's influx of antigen-specific immune cells. So our hypothesis was within these skin lesions, you're naturally increasing the number of pathogen-specific cells. So when we take that biopsy from that little piece of flesh, again, there's bacteria present and these T cells, all we do is grow those T cells out from that piece of skin tissue and then expand them. So we grow them out and just get as many as we can. And then that, again, polyclonal CD4 T cell line is what we used further downstream to test for specificity against different treponema pallidum antigens.
That makes sense because it's much more concentrated, especially if you have an active infection going on in the skin.
Correct.
And I can see why you'd want to go back and take a look at those with a primary lesion as well, because I think that would be really interesting findings.
Mm-hmm.
How did you find or identify the T pallidum antigens?
That's a really great question. So I'll preface this by saying that treponema pallidum has a relatively small genome for a bacterium, but it does have over 1,000 predicted proteins. So there's over 1,000. We expressed each of these proteins individually, like one at a time, but it would have taken us a long time to do all 1,039 proteins. So we started off by targeting our expression of these different proteins to known outer membrane proteins.
Because they're protective.
Because they're protective, potentially. We also targeted things that we knew were highly expressed, either during in vitro culture or during active, like in vivo infection. And then we also included proteins that were known to be B cell antigens or targets of antibodies during rabbit experimental infection and then also human infections. So this comprised our list of goal proteins to express. We didn't express everything, either because of different experimental challenges, but at the end of the day, we ended up with 89 proteins that we could use in this library of proteins to test and look for CD4 T cell responses. At the end of the day, it's less than 10% of the entire proteome, but we tried to use a rational approach to get started.
And how did you express these proteins?
So we use a cell-free E. coli expression system. So it's nice because without all of the entirety of E. coli, we're able to express these proteins without LPS, which is important for these in vitro T cell cultures.
And is it because LPS can potentially activate the T cells?
Potentially, but also for PBMCs, because PBMCs are such a complex mix of different cell types, the LPS could stimulate different immune responses in that setting as well.
And for our audience, LPS stands for lipopolysaccharide.
Correct.
And so what did you find when you did these experiments? Which T pallidum proteins were recognized by the CD4 T cells? And you can separate it out in terms of those from the blood and those from the tissue if they're different.
So in some, we included 8 patients in this initial study and found CD4 T cell responses to 14 different treponema pallidum proteins.
Out of the 89 that you had started with.
Correct.
Mm-hmm.
Those included outer membrane proteins because we made an effort to include those in this study. So things like our lipid transporters that are in the outer membrane, so presumably essential to the treponeme. Also responses to a group of proteins called treponema pallidum repeat proteins, which have a lot of different implications. We also found CD4 T cell responses to a highly reported on protein called BAM-A that's also known to be a target for antibody responses. In addition to these things, so these are kind of proteins on the outer membrane, including a fibronectin protein, fibronectin binding protein. We found CD4 T cell responses to proteins that are under the outer membrane. So things that would not be exposed to the host immune system from an intact, alive spirochete. So those include some of our diagnostic proteins. So some of those proteins that are used in our treponemal tests. And then we did detect a response to a single cytoplasmic protein.
So we know that there's different strains of T. pallidum. How different are the strains in terms of the proteins that you're talking about? And do we have to think about that variable when you're thinking about the immune response?
Yes. So that's a really juicy question. So there are strain differences, and there are differences amongst strains in some of the proteins that we included in our library. So one would say, well, geez, Tara, did you include multiple types of each protein to represent all these different strains? We did not do that initially. We included proteins that represented the nickel strain. So this is our laboratory strain that we've had circulating and propagated in rabbits or in culture for, I think it's over 130 years now, maybe 120 years. But we started there. My rationale for doing that was that it was shown in other highly variable proteins, TPRK as an example, that the variable regions of the protein consisted of B cell epitopes, whereas the conserved regions of the protein contained the T cell epitopes. So extrapolating from that, we presumed that the T cell epitopes, the things that we're after, would be located in the conserved portions of these proteins compared to the B cell epitopes.
And did you find any difference in the recognized T. pallidum proteins, for example, in those who had a history of syphilis or, for example, those who were asymptomatic?
So that's a really great question. I really want to answer that during my career, but we had a limited number of patients enroll in this initial study. So
it's 8 patients, right?
8 patients, right? So it's a really small number, and I'm not powered to answer that question.
So maybe a future question.
Yeah, in the future. So the other complicating fact is when we diagnose an asymptomatic infection or symptomatic, sometimes that's not entirely clear. If somebody is diagnosed as having early latent syphilis, is it that they truly have no lesions in their body, or that we just didn't look hard enough to find that primary chancre?
That's a very good point. And they have found that a lot of people diagnosed with early latent syphilis might actually have had primary or secondary that got missed on exam.
And then another level there is if I'm saying that somebody has early latent syphilis versus late latent syphilis, is late latent truly late latent, or is it syphilis of unknown duration and we just don't have the timely serologic data to distinguish these two things?
Yeah, that's a great point. And do you think the immune response is durable and or protective? I know you only had 8 patients. Are you planning on following these patients over time?
To answer the first portion of your question, do I think it's durable? Yes, I think that these immune responses are durable. And I think that because we were able to enroll some patients that had previously treated syphilis, right? So when we took their PBMCs, when we took their blood, they did not have syphilis. They were treated 3 years ago, 5 years ago, one person 10 years prior to coming to our study. And we were still able to find these circulating CD4 T cell responses. In addition to that, one of our participants who had secondary syphilis, we were able to detect skin CD4 T cell responses. And then even after treatment, 6 months afterwards, we're still able to find these Treponema pallidum specific CD4 T cells in their healed normal skin. These things together make me feel confident in saying that, yes, these are indeed durable responses. Your next portion to the question was, how protective is this? Again, we don't know yet. I will say that some of our participants do come back to this study, either because they were reinfected or to be enrolled in a different cohort. And just very, very anecdotally, we were able to identify at least one case of somebody who was initially enrolled of symptomatic syphilis, was treated, living their life, happy, and then re-enrolled with early latent syphilis. And we were able to detect differences in their CD4 responses between those 2 episodes of syphilis. So again, this is an N of 1, very anecdotal, but interesting.
It is very interesting that you did have a patient who was reinfected with syphilis during your study and you were able to see a difference in immune response. And was this a difference in terms of the number or type of proteins the person had CD4 T cell responses to?
Yeah, so in this situation, the person did have Treponema pallidum specific CD4 T cell responses, but with our admittedly limited library of proteins, we weren't able to identify CD4 T cell responses to any particular antigen. So none of those 89 that we included. But on re-enrollment, we were able to detect within that panel of 89 proteins, CD4 T cell responses against particular antigens. So there's a difference there, right? We weren't able to detect anything, again, with our 89 protein panel. But later, after reinfection, we were. So what does that mean? We're not certain yet, but there was a difference.
Do you think that the immune response would change in someone with HIV, for example, someone who had a low CD4 T cell count or who was not on antiretroviral therapy?
Yes, I really do think that there would be a difference. It's hard to say if there's just a difference in the robustness of the CD4 T cell response because there may be fewer overall CD4 T cells in the body versus a different breadth of response. By that, I mean, it's unclear to us currently whether active HIV infection with a low CD4 count means that people aren't responding to as many antigens, or if their responses are just muted across the board because the total number of CD4 T cells is low. So it kind of gets at the question of whether it's a sheer numbers game or if there's just the general CD4 T cell dysfunction that might impair the response against Treponema pallidum. Clinically, we know that this is something that happens, that people living with HIV that have uncontrolled viral replication, they've got low CD4 counts, they are at increased risk of developing neurosyphilis. They're more likely to have atypical presentations of their syphilis. And importantly, this is the thing that I think about the most. We're much, much, much more likely to present with malignant syphilis or Louis maligna, which is a really, really striking presentation of Treponema pallidum infection.
And it probably depends on also where their CD4 count is. Like, for example, if their CD4 count is 10 versus their CD4 count is 190,
right,
that might have a different response. And whether or not they're on antiretroviral therapy can also influence that as well. Tell us a little bit about HLA class restriction and how did you determine this?
So we were focused on CD4 T cell responses. These CD4 T cell responses are dictated by the CD4 T cell receptor, so the TCR receptor, and how that T cell receptor interacts not only with an antigen, but also with the MHC class II molecule on antigen-presenting cells.
Immunology 101. This is great.
MHC class II, that is our major histocompatibility complex 2, which also we discuss as our HLA type. So if we're reliant on our HLA type to bind specific antigens and interact with the TCR,
the T cell receptor, it is important to understand the T cell class restriction to understand the antigens that we're detecting. Another way to say that is somebody with one particular HLA type might respond to T pallidum differently than another individual with a different HLA type. So although we might have detected responses to these 14 proteins in this particular group of individuals, another group of individuals with a different HLA type might respond totally differently, and maybe we won't detect those same 14. So it's very important thinking about the immune response.
Exactly. Also super important if we're trying to generalize these CD4 T cell responses to bigger populations. So everybody in the United States, everybody in the world, because if we intentionally include a CD4 T cell antigen into a vaccine, we want that antigen to be recognized by the majority of people.
What are some next steps in your research in this area? And what are you planning on doing?
Oh, so exciting.
The CD8s?
Yes. So, well, first I'll say for next steps, just as you were asking about this difference in asymptomatic versus symptomatic reinfection, all of these things, we're hoping to continue enrolling to get our N up and be able to have enough people to say whether there's a difference in the CD4 T cell response between people that have a symptomatic reinfection versus an asymptomatic reinfection, such as early latent syphilis. So that's one exciting thing that I want to get at. Another exciting avenue for this research to branch out into is understanding not only the CD4 T cell response, but also the CD8 T cell response, which is a huge black box. We really know almost nothing. And so I'm excited to get into that CD8 realm and better define that.
And are you able to do some of the similar laboratory techniques to sort on CD8 T cells to do the same types of experiments?
So yes, we're able to isolate CD8 T cells that we think are T pallidum specific, but to test for reactivity to particular antigens, it's a little bit trickier than it is for CD4 T cells. So CD8 T cells, as you might remember, tend to respond to intracellular antigens. So these are antigens that are presented to the immune system by very specialized antigen-presenting cells, and then also other cells in the body in the context of MHC class I. So to do that experimentally in the lab is a little bit different. And so we're working on understanding those pathways for antigen presentation, because it is possible that T. pallidum, one, has some portion of its life cycle within cells, maybe less likely, but we don't know. Or two, there is a cross-presentation mechanism that's important for presentation of T. pallidum antigens in the context of MHC class 1. Another interesting thing to think about for these CD8 T cells is whether or not they are indeed antigen-specific, or if they're more on the innate-like spectrum, meaning that maybe they're responding to more generalizable bacterial antigens versus T. pallidum specific, like proteins.
This is very exciting. I look forward to hearing more and seeing what results you end up having as your experiment continues over time.
Yeah, well, thank you. I'm excited to get into it.
What is the most common question you get on this topic in terms of the immune response to syphilis or syphilis vaccines?
It depends on who I'm talking to. I would say that the most common question I get from people that aren't used to thinking about syphilis or syphilis immunology is, why don't we have a vaccine yet? So that's a very common thing. And so that it's multifactorial, right? But we're working on it. I think that there's more and more people here in the US and abroad that are actively working on these questions. And it's actually a very, very exciting time to be a syphilis immunologist. Amongst my friends who love to talk and think about syphilis, I think the question about the CD8 T cells is really common.
We appreciate you being on this episode, Tara. It was fantastic, and I learned so much from you. Thank you for being here and for your time and discussing the immune response to syphilis. We really appreciate you.
Thank you. Thank you for all the work that you do.
This podcast is brought to you by the National STD Curriculum, the University of Washington STD Prevention Training Center, and is funded by the Centers for Disease Control and Prevention. Transcripts and references for this podcast series can be found on our website, the National STD Curriculum, at www.std.uw.edu. Thank you for listening and have a wonderful day.